Click for IB Biology. Here we're looking at the movement of water in plant tissue. In video 2 we looked at the movement of water in animal tissues. So now we move on to look at the plants. There's a blank PDF of this presentation down in the description underneath, where you'll also find a quiz with 10 questions and the answers. So you can make your own notes and test your own understanding using those resources. So let's take a look at water movement in plants. So the first thing to do is to take a bit of a reminder about the structure of plants. And of course, we have in common with the animal cell, we have the cell membrane and the movement of water across that cell membrane. So that's the controlling feature. We know that within the plant cell, there's also a vacuole, which is often used as a storage area for sugars and amino acids, and so that may well have a big influence on the movement of water and the concentrations, the solute concentrations that we encounter. The cell wall, which goes around the outside of the cell membrane, should be considered as fully permeable to water. Now, the cell wall is very important in this topic, but it doesn't act as a barrier to the movement of water. And so in each of the images that we can see here of plants, we see that the cell wall features prominently within structures. And the kinds of topics where the movement of plant cells is going to become important to you as a biology student are in the idea of support of plant tissues. Without a skeletal system, how do they achieve it? The movement of water in roots, the uptake of water and transport to the leaves, the movement of sap around the plants, and the opening and closing of stomatal pores. There are other aspects that are important, but those are some of the ones that we're going to encounter quickly on this course. So here's our definition again. Water moves from the low solute concentration to the high solute concentration. And we know that the reason for that is because over here we've got a higher kinetic energy of the water molecules, total kinetic energy, and over here it's a lower total kinetic energy of water molecules. So they're moving more on the left than they are on the right. So let's take a look at hypotonic and then hypertonic. So hypotonic in a plant means that the external solution has the low solute concentration, and the cytoplasm and vacuole by comparison has the high solute concentration. So it's low to high in terms of the water movement, and so the water is going to enter the cell. As before, the volume of the cytoplasm will increase. But what's going to happen this time is that as the volume of the cytoplasm increases, the expansion of the tissue will be resisted by the wall pressing back in the opposite direction. And so the plant will experience a pressure increase. This pressure increase is called turgidity. And turgidity in plant tissues is what gives the whole plant support. So with lots of water, the cells become turgid. There's an outpressing of the cytoplasm. The wall pressure generates, and this is what creates the well-watered and well-supported plant. Now let's contrast that with plants in a hypertonic environment. So within a hypertonic environment, the outside of the cell has the higher solute concentration. The cell contents, the cytoplasm and the vacuole, have the lower solute concentration. So we know that what's going to happen is the water molecules are going to move out. The consequence of this is that the volume will decrease. The pressure which came from the walls is going to decrease. And so the plasma membrane, or the cell membrane, it actually detaches from the cell wall and creates these spaces around the plant like this. So the cell membrane is shrinking inward, and so the pressure has been lost. In plants, this phenomena is called plasmolysis, and that's how we describe it in tissues. In the whole plant, we would describe this as wilting. This is where a plant, which is well watered like this, becomes plasmolized in a hypertonic solution, and the plant, this plant here, the stem will lose its form, and the leaves will lose their form as well to become wilted like this. And we know that we need to water our plants when that happens. So this hypotonic situation seems to be the one which plants desire, unlike the isotonic of animals. So hypertonic is not desirable at all. Isotonic is, in contrast, isotonic is not as significant in plants because at that point the amount of water going into and out of the cell is roughly equal, and at this point the cell is on the verge, just about to become plasmolized, and in fact we call that incipient plasmolysis. Now, from the point of view of the natural behavior of the plant, this is of no great significance. The plant wishes to be hypotonic. However, isotonic environments do allow us to carry out experimental techniques in which we can determine the concentration of the cytoplasm of a cell. And you may have experienced this practical in your laboratory on your course. But let me just outline in broad terms what we're going to do. What we want to find out is if we have a potato, the question is, what is the concentration of the cytoplasm of a potato? And this method requires that we create different concentrations of external solution. So the external solutions are here, and so down here we have the dilute ones, and they will become increasingly concentrated as we go along. And we take a sample of potato and we put that into the different concentrations of fluid, so different pieces of potato. And what we're interested in is one way of doing this is to look at the percentage change in mass of the potato. Now, with very dilute, what we would predict is that the water will go into the cell, and so we would expect that there will be a positive increase in mass. But as the concentration of the external fluids increased, then the percentage change in mass will decrease. And in fact, once we will actually get to a point where the external fluid is actually hypertonic to the cell, it will begin to lose fluid like this. Now, to get this technique to work, you will have to do many repetitions of this particular one here, but then so that we could actually plot the mean and perhaps things like standard deviations. And then by taking a line, we can draw, sorry, that wasn't very good, but you understand what I'm doing. We can see where it cuts the line here, which is where there's 0% change in mass. So if we have a potato cell and the amount of water going in is equal to the amount of water going out, then there will be 0 mass change. So we know that this concentration on this axis here, we know that this concentration is the concentration of our potato cytoplasm. I'm sure you'll explore that technique in your laboratories, but that's just a little bit of background theory on the isotonic nature of that. There's a link to the next video, to the channel, and to the playlist.